Swayam Prabha. Digital India. Educated India.
Hi everyone, welcome again to the course of structural biology. We are at the end of the module visualization, and in the last class I talked about the visualization platforms, the platforms which are used for looking at the macromolecules, analyzing them and making publication quality figures. I continued with one of the example visualization platform, which is Pymol. I have demonstrated the initial properties, what you could do from uploading the PDB molecule to analyzing it, and today I will continue from the part I have finished. So, let us continue with the demonstration of the 3D visualization using Pymol. One of the critical part of analyzing a macromolecule is to view the side chains. Why side chains are important? If you remember the protein structure which is built up from amino acids, there are 20 different type of amino acids and 20 different side chains are there. The side chains gives you the properties. So, we need to look at them for our analysis. So, when you want to choose the residues, and you have to type the following command: set cartoon underscore side underscore chain underscore helper 1. Then you could view the desired residues by selecting them and choosing so rather than sticks. For the selections I have written the command, so you could follow it here. So, you see you could have look at one side chain from here, and then you could also, for better viewing, hide the main chain and only show the side chain. For glycine, if you remember, the side chain is H, so you have to put the following command, and with that I want to show you the demonstration. So, now you see the screen. Whenever the screen comes and you start, you have to load the PDB file. Here I have loaded the PDB file of the enzyme called beta lactamase from Mycobacterium tuberculosis, which I discussed in the CUT session, and now you have to select the amino acid for which you have want to show the side chain. Here we have chosen the serine, which is serine 84. This is the catalytic residue of the enzyme. Through this enzyme, the enzyme perform its catalytic activity, and you see that the serine is here represented, the whole protein, the whole enzyme is represented in cartoon format, whereas we have given a different stick format to the amino acid. This will help you to identify the amino acid, and next also this will further help you to identify the interaction of this catalytic amino acid with other ones. Now, if you remember, I talked about there are 20 amino acids. All of these amino acid have side chain, whereas glycine have only hydrogen. That makes glycine a achiral amino acid, and here we will show you how the representation of glycine happened. You see here glycine is selected, you will see the selection, and then again the stick format is given here, so that you could see the glycine. And now this glycine, if you see, is only having hydrogen, and that is why it is a special amino acid to mention. So, coming back from where we start, we talked about side chain. Now, we will get two amino acids, their side chains, and then it would be utilized to measure the distance between those two residues. So, if you see in the picture, you will see that there are two amino acids and their distance is first identified and then calculated. Distance will help you to understand there is a non-covalent bond formation or not. So, wizard to the measurement, what is happening? You have to click on the two atoms whose distance you want to measure. Now, you have to remember that do not keep the distance label from Pymol in your diagram, label the distances separately in the PowerPoint. Why? Because you measure it, but you do not label it. If you label it in the PowerPoint, then you would have control of changing them whenever you want. Change the dash color to any color of your choice, you see the dashed. So, usually we present in Ruby color, use the following command: set underscore color name, red gap float, green float, blue float, which is the color RGB code. So, with this I will again switch to Pymol and I will show you. So, again this is the window. You have remember that we have already selected the serine. Now, what we are selecting is lysine. So, when you are selecting a lysine, you know that you get the nitrogen, and we will check that what is the distance between the serine and the lysines, oxygen and nitrogen. So, when you do that, you could see that in this case the distance is 3 angstrom, which clearly shows that these two amino acids are in non-covalent interaction, which is hydrogen bonds. So, and if you remember, I talked about, I am repeating again, you will not take the name or the colors which are given here, you go back and do it in the PowerPoint. So, you could make it a ruby colored, generally default, but do not put the distance. So, this is the interaction between the serine, the catalytic residue, and the lysine, the secondary catalytic residue, which helps in the catalysis by taking the proton from the serine, making the serine O minus. Coming back, we will show how to save an image. So, you go to display, then you go to background, and then you make it white. If you remember, I have already done it, because as I told you, whenever you want to save an image, you want to save it in white background. So, when you take a printout, you do not spend unnecessary ink. Then you have to type the following command in the terminal: ray 1080720, or choose 600 dpi from the ray drop down box on the right hand side of the Pymol window. You have two options, and then uncheck the transparent background option. Then file, save image as PNG. If you want to save it in PNG, there are other options, and then save. Also, if you are careful, you would like to save the .pse file of your structure, because if you do that, for any future reference regarding the interaction you want to make an image, you could change it. So, that is always clever. To save the .pse file, file, save session, save. If you remember, I have shown you that option, and this is the ray option. If you remember again, in the POB ray, in the raster 3D, those are the technology which come from them, and now it is integrated to any visualization platforms. So, with that I would again switch and show you the condition which we have in the Pymol. So, as I talked about, we want to save it. So, if you see now you change the parameters, the DPI values and all, so that you could make a high resolution picture. See the development of formation of the rendering is happening. Rendering means if you carefully see the screen, the picture is getting sharper at the edges, becoming better. See now you get this portion, it is already selected the part you want to save. So, you put any name on that and you could save it. So, you save it. Also, as I told, at the same time you go to file and you save the session. You put save session or save session as, and then you save it. So, you name, and as I told, it would be saved in the format of .psc or .pze. These are the two format which save your session. Coming back again, so we talk about protein, but a protein or enzyme, the enzyme beta lactamase which is there, we are talking about the interaction of protein or enzyme with its ligand, with its substrates are important to study. So, for doing the study you have to select the ligand from the protein sequence. You have to go to the selection which is select tab and choose show as sticks, so that your protein would be represented in cartoon. If you see, your protein is represented in cartoon here, and then your ligand would be presented as sticks, ball sticks are the options to generally represent the ligand molecule. After that you choose show spheres, then in the command prompt type and execute the following commands one by one. Set sphere underscore scale 0.25 Sile. So, what you are doing, you are making the size of the stick and sphere optimized. Set underscore bond stick underscore radius 0.2 Sile. Make sure you have selected the ligand before executing these commands, because if you do not select the ligand, it would be applicable to every bonds, right. The only way you could differentiate between the ligand with others is by selecting it and then selecting the parts. So, let us again go back and look how it is possible. So, we delete the previous one and we are now loading a new structure. See, there are so many PDB files and you choose a file. See here, the chosen file is shown. If you see, the guys which are in red sphere are phosphates here, which come through the condition of the crystallization, because this is a structure which is solved by technique of protein crystallography. Now, you could remove water or you could keep them, you could remove the phosphate, you could keep them. So, all these things whatever, here we remove all of them, so that the structure become clean, and you see the only thing which is attached with the protein is the ligand. This is again the same protein, the beta lactamase from Mycobacterium tuberculosis, and the drug is tebipenem. So, you could select the drug as I told, and you could add those comments so that you could make the ligand more easy to view. See, if you put everyone's sphere, it will look like that. So, you have to be careful at each stage, and that is the only way you could develop a good figure. Also, if you take a look carefully, you could remember through quote I have already shown you the two different tebipenem conformation, tebipenem structure with the beta-lactamase. Here you will see that the 4-membered ring is present or absent, and the unavailability means it is making a covalent bond, which is the normal case where the acylation reaction is happened. Now, you see the representation of the ligand with the ball and the stick. Going back to my presentation showing active site residues. So, although we have already shown the residues of serine and glycine, these are the active site. So, every time what you have to do, you have to open the protein PDB file in Pymol, then select the active site residues. How you select it? It is a very interesting question. A lot of beginners who come and take training with me, they ask me. So, there is no rule. Generally, we go to the literature and read about the protein, which gives us the knowledge about the active site. But if you are becoming expert, then you could trace the substrate, the ligand just now we have discussed about. You could go back, you could trace, you could see the interaction, and you could identify who are the amino acids which are making the interactions with the substrate, and they are the active site residues. So, under selection tab you have to choose show, then sticks, color by element, choose color of your choice, and then type the following command in the command prompt: set cartoon underscore side underscore chain underscore helper 1, and then as I always say, label that in PowerPoint. So, you will get a thing like that. You see serine 70, serine lysine 73 and glutamate 166. When we will go back, we will show you that residues, but different numbering. If you remember, I told these are the numbers which are standard, but each of the protein, each of the enzyme from every organism have their own numbering. So, let us go back and see the numbering here. Here the serine is 84, and then we go to glutamate, which is 182, and then we will show lysine, which is 87. So, if you see, these are the 3 amino acids which perform the 2 reaction I talked about in the COOT class, which involve acylation and deacylation reaction. Here you see the lysine have taken the proton from the serine, and the serine attack the beta-lactam ring and form a covalent complex, what I have talked about. So, now you know this is the active site and how those active site residue are closer to the substrate, you would see the periphery, right. Let us go back. Viewing an oligomer. I have already shown you a dimer, but here we will come with a different type of representation. You see the oligomers, but here you will see some different oligomers to show their property. So, as usual, you have to open the protein in Pymol, Then you have to do right click on the protein molecule in the viewing area and you have to choose chain, color, by chain, then by chain. So, when you do that, you will get this dimer. You see the pink and green color representing each of the monomer. So, again before going into the monomer, form a multimer, go back and check it in the Pymol. So, first you have to clean it everything and then you have to load a oligomer. So, you see there is a dimer, but you could only be confirmed when you go to color and color by chain. Yeah, two colors. So, that means it is a dimer. So, we will come again. So, it is clear to you now that this is a dimer. So, again for how they are forming, how you could select monomer, again you have to open the protein. If it is not open, in our case it is already open, then right click on the protein molecule in the viewing area and then you choose chain, extract object. The selected chain would be created as a separate object which you could name as obt01 by default, but you could have name it of your own. So, there is the dimer and it would be a monomer which would be a result. Remember, I told you, you should always make a monomer before going to do the alignment. So, let us see how to make the monomer here. The good part is now the commands are written here, so you could easily follow them. So, you extract object, see I have shown you in a different way. Now, this is a different way to make it. So, see you have a monomer now. So, this is the way you always convert the oligomers to monomers. Now, what to do with the monomers? You could do alignment with other proteins, alignment with the two monomers. To do that, again you have to open the two proteins, or if one protein is loaded, you have to open the second protein. So, you have to choose one protein as the template and to align the other protein with the template, go to this proteins tab and choose action, which is the A. If you remember the 5 things: action, show, hide, label, color. So, A, then you go to align, then to molecule, then choose the template protein. So, this is an example of alignment and you see that these two proteins are looking at the alignment, you could see that these two proteins are same or having identical fold with very high identity. So, let us switch and see in the Pymol window how we could see the alignment. So, first as usual you have to open the PDB file, one PDB is there, then you have to open other file. So, second protein is there and then you have to do the alignment. So, as you remember, you have done the alignment and it shows the RMSD value 0.727 with 177 atoms, which means the alignment is quite good. So, now making mutants. For mutants, you could make mutants in Pymol, but as I told, for modification we believe we trust more on the other type of softwares. So, we do it in WinCut. So, what to do in WinCut? You open the protein PDB file. I hope you remember, I have given the demonstration. You go back and you see draw, you go to draw and then go to atom, click on the drop down triangle next to the chain A and choose the residue that you want to mutate and then click on apply. The chosen residue would be shown on the menu shown on the right hand side of the window. Click simple mutate button if you do not have the electron density map. So, simple mutate, no ED map. But when you have ED map, the mutation would be adjusted according to ED map. So, there are two options. The mouse pointer changes to a plus sign. Click on the residue that you want to mutate and choose the amino acid. So, when you do that, if you remember, you will get a list of all possible amino acids. Now, you choose the amino acid you want to change it to, then go to file, save coordinate and save this PDB file of mutated protein. So, you have done mutation to your protein. Now, you want to view the mutated file in Pymol. Again, you open the protein wild type and mutated PDB files. You align the two structures keeping wild type structure as the template. You select the residue that shows the mutation in both the sequences. Under section tab, choose show sticks. So, you make them in sticks. Color by LM, choose color of choice. Unselect the mutated PDB file and perform ray for the wild type structure and save the image. So, here you see the residue was glutamine 39 and you make mutation to this glutamine to lysine to make it lysine 39. So, that is something you have mutated it already in CUT and now you see it in Pymol and you save it in high resolution for publication. So, to demonstrate that, we again come back to the screen and now we will see the mutants. We will also compare between the wild type and the mutant, so that we could have a comparable picture. And as I told, we could have made the picture in CUT. Why we did not do it? Because when we do that in CUT, we cannot get a nice picture, a high resolution picture. So, here we are making a mutation, like finding a mutation. The serine 84, the catalytic residue is mutated to alanine. So, in one side there is serine, in other side there is alanine. You clearly see the difference, they are same protein, but serine in place of serine, alanine is observed. Now, you have to select them, you have to make the stick representation and now you will see the difference. So, in one place you have the serine, in other place you have the alanine. You could easily see the absence of oxygen. CH2OH is serine, CH3 is alanine. Coming back to, so again here what I forget to tell you, would save the session for future reference and you now take them to the PowerPoint and make the figure according to how you like to have them. Selection of loop. Loops are playing very critical role in function, right. Here you see a big loop and if you see kind of carefully, you would see there is a omega type of representation. So, it is called omega loop. This loop is a characteristic loop in, especially in class A beta lactamase, though in other class of beta lactamase also it is found. So, what you have to do? You have to select the loop residues in the protein. In the select tab, you have to choose color by LM, choose color of choice, then click on action, copy to object, new. A new object by default obj 0 1 would be formed by default, or you could have given your name. In the obj 01 tab, choose show sticks, then you would have create these two images and label them in PowerPoint. So, again go back and take a look how we are taking the omega loop as I was talking about and selecting it. So, we have to keep it in cartoon and then you have to select the omega loop. You have to know the numbering which represent the loop, and then you have to work on selecting the loop, and by doing that you have to know where the loop residues are, and then you have to again color it by element. So, you will see that a nice omega loop is there. Then you have to represent them by stick by making a separate object, as I told. When you do that, you could save it as obj 01. This is the loop, and the molecular representation will describe about the composition of the amino acids which are present in the loop. Also, you see there is a 310 helix which is part of the omega loop. Again I am switching. So, if you remember, I have shown you the differential binding of the tebipenem in two structures. This is because in one there is a mutation, as I have shown you earlier. The mutation is lysine to alanine, and because of this mutation the lysine could not have taken the proton from the serine could not react. So, two Blasey structure we have, both are with tebipenem, but in one the tebipenem could not react where the mutation of lysine to alanine happened, and one where reaction, the first step, the acylation happened. So, that is the scenario. To do that, first you have to show the protein in cartoon mode, then choose the ligand from the sequence and show it as sticks, then choose the ligand and click on show sphere, because you remember we want to show the tebipenem in ball and stick mode. Select the ligand and reduce the radius by typing this in the command prompt. Set sphere underscore scale 0.25 cell. You are fixing the sphere, then choose the ligand color whatever you want. I want to show you that. So, I switch here again you have to delete the object, you have to load both the tebipenem complexes, teb mm and teb Ca. Teb Ca is already loaded. So, another one is teb mm. So, the two Blasey structure, the same structure with two state of the carbapenem drug tebipenem. So, we have made them here. If we align them, then it would be easier for us to take a look. We could also remove the other parts, the water, the phosphates, and then do alignment. So, when you see the alignment, you see how good the alignment is. Now, we could probably make them, bring them in the center, so that you could see the two tebipenems. Remember, when we have seen these tebipenems in Coot, we have already know that they are differentially bound, but now bringing them in Pymol, the pictures would be much better. As you see, you could have focus it, orient it, and represent a much better structure than what you have seen in Coot. So, here you see the two tebipenems. I am switching. Protein alignment, I have shown you the alignment. If the proteins have good fold matching, they have low RMSD value. If the matching is not great, then the RMSD value would be high. As you see here, RMSD 0.866 when the two proteins have nearly identical fold and sequence, whereas when it is not, the RMSD is 14.978. So, as usual, you have to first show the proteins to be aligned in cartoon mode, then action align to a molecule, select the object, then you have to put the command align moving structure name target structure. So, align first the structure which have to be moving and then the structure which is being steady. So, first the moving structure, where moving structure is a PDB which needs to align with the target structure. So, I will go to the Pymol window and I will show you the alignment. So, you have to load the PDB file as I am doing here, and then you need two PDB file because you have to do alignment. So, there are two PDB files, and when you are doing the alignment between them, you will see that align to molecule, you will see good alignment and RMSD value is 0.727, means they are very good as two close structure. And now we will see another alignment where we are trying to align two different structures, and when you do that you see the RMSD is 14.337, which is clearly showing that the two structure have very less fold similarity. So, understanding this, comparing structures is a very utility tool. Now, coming to find polar contacts, as I told, when you want to understand the deeper level of science, you have to find out the interactions inside the protein, what are the interaction with the protein with the ligand and all these things, and that is called finding polar contacts. First you have to select the ligand with whom you want to see the interactions, then go to action, find polar contacts to other atom in the object, and it is essential for finding hydrogen bonds between ligand and a protein or two proteins, between two proteins or between two objects. So, let us see the Pymol demonstration of finding polar contacts. So, again you have to load the PDB file, and once you are loading the PDB files, what you have to do, you get the polar contacts. So, if you see here, you have identified the polar contacts with tebipenem. Tebipenem is a carbapenem, as I told. So, here you go and you find polar contact to the other atoms in the object. Here you see the yellow lines are indicating the possible hydrogen bonds which could form between the protein and the ligand, also the water molecules which are present in the protein, means which are catalytically important. So, now you did them, you color them, you come back to PowerPoint and you label them. So, we have shown in these two classes how to get a lot of different things, how to do that through Pymol. But one of the extremely critical things through Pymol is to make good figure. Probably in these two classes I have told about this for multiple times. So, now this is time we are going and showing you how you could make publication quality figures using this software. So, what are the software used? You would require the following softwares: Pymol, which is the major software, WinCode, which will be helping you to guide, and Microsoft PowerPoint, which will help you getting more control on the structure. If you use the labeling coming through the softwares, then you lose your control. You probably want to show some interaction, or you do not want to show, you will not get those facilities, you will not get those advantages if you allow the Pymol software by itself to label. First of all, you could make simple figure. For making a simple figure, you open Pymol and view the required residues. You choose a particular color for your enzyme, and like here we have chosen the shade of green for the enzyme TM, and you remember to follow the same color scheme for all of your figures, so that you make a continuity. Like you have two proteins and you make through two different color, it is helpful to make the signs visible. The main structure is viewed as cartoon and the residues are viewed as sticks. The ligand is shown as a ball and stick model with ball radius 0.25. After viewing, save your image in proper resolution. The diagrams are always saved in white background, as I have talked about. Then open the saved image in Microsoft PowerPoint, insert text boxes and label the residues, keep the font as Time New Roman and fix the font size of the residue to be to 14 and of the ligand to be of 11. Bolden the residue and the ligand name and keep the residue number in the subscript. When done with all the labeling, press Ctrl A and right click on the pic. Choose save as picture option and give a desired name and save it at a desired location. So, this is one of the high resolution picture developed you could see. So, you could also follow the scheme and you could make a simple figure. Zoomed figures. So, you have one mutation or one residue, you make a mutation, you want to show the environment zooming it. So, make the required sample figures needed to make this compound diagram following the steps mentioned in the previous slide. So, following that you could make the simple figure. Once the simple figure are ready, now you open them in Microsoft PowerPoint and set as desired. So, here you are actually not creating an image, you are mixing few image to make a complicated image which actually explain the science you want to project. To show zooming in of a region, insert the desired shapes to show that. Then here you see we have used rectangle shapes, you could use rectangle shape, you could use any other shapes. Set the shape transparency to 100%, set the line color of your interest, like here we have chosen red. Set the line style to dashed and choose the desired line width. So, these are all not needed scientifically, but more carefully doing that you will make more attracting publication quality figure. Set the line style to dashed and choose the desired line width. We have chosen 3-point line width here. You always remember to follow the same condition once chosen for all your diagrams. A continuity again show the maturity of the author who is writing the manuscript and creating the figures, or describing a project as a undergrad, as a masters you do projects. When you are representing the project result, if you make continuity in figure making, that shows how good you are, how mature you are. To show zooming in, click on insert shapes line. Follow the same thickness and color conditions for the lines as per the shape chosen. Once done, press Ctrl A and click on save as picture and save as desired in the desired location. Important Pymol commands used, I have made a list. To open a PDB file, I have shown you open Pymol terminal and type the following commands: fetch PDB ID to the command line. If the structure is already saved in your computer, then you can follow the following: file, open, choose the desired file from the desired location and then click open. To measure distance between atoms, wizard measurement, again we have shown that. Click on the two atoms whose distance you want to measure. Do not keep the distance label from the Pymol, as I talked earlier. Label the distance separately in PowerPoint, change the color, and then in Pymol terminal choose the ray option and use the following command to set the color and set the dash options. To set a particular RGB color, open Pymol terminal and type the following commands to set a particular RGB color. Set underscore color, name, you have float, red, green, blue, RGB. After this command, type the next command as follows: set cartoon underscore color, provide the name. For example, as we have shown earlier, you use hay color, which is 01020. To view only the side chain of a residue, set cartoon underscore side underscore chain underscore helper 1. So, these I have listed them, though I have demonstrated, but I have listed them again so that you do not make a mistake, and all these are quite organized properly. Then view the desired residues by selecting them and choosing show sticks for the selection. To view the main chain of a selected residue, set cartoon underscore side underscore chain underscore helper 0 cell A. To set sphere radius of ligand, open Pymol terminal and type the following command to set the ball and stick radius of the spheres in ligand. Set sphere underscore scale 0.25 cell A. Make sure the ligand is selected for using this command. To set stick radius of residues, set underscore bond stick underscore radius 0.2 cell A. Make sure the residue is selected for using this command. Lastly, to save an image, open Pymol terminal and type the following command: display background white. You remember I told repeatedly that you have to make your terminal white. Then type the following command in the terminal: ray 1080720, then file, save image as PNG save. Also save the .psc file, as I again talk about this, that .psc file helps you to bring the same thing again. To save it, file, save session, save. So, the summary: if you have TEM, which is a beta lactamase, we have shown here TEM cartoon in RGB color, you have to use 01020. You could choose your own enzyme color, stick radius of residue when also showing ligand 0.2, ligand ball radius 0.25, water sphere radius 0.2, dash RGB color ruby. Labeling values, it is a list of all the ratios used for making and labeling zoom diagrams. All fonts Time New Romans, residue name font size 14, ligand name font size 11, residue name font style bold, ligand name font style bold, residue number subscript, shape line thickness 3 point, shape line color red, shape line style dashed, shape transparency 100%, distant label size 11, distant label style bold. So, is it mandatory to do that? No, there is nothing to be fixed, but we have given you those numbers. If you apply, you will be very surprised and very elated to see that you have made a beautiful figure, publication quality figure out of Pymol. Once you do that, then you make all changes according to how you like. You might not like, the font of Times New Roman, and all these things you could do, but the ratios are important. So, with this I hope you could learn more. As I told in time of COOT, I am repeating this: Pymol, COOT and all the related softwares like VMD, Chimera, they are coming up with thousands of choices. How to know them? Just to play. I have given all the links. Download this software, get the PDB file from protein data bank and do anything and everything. More you play, more you spend time, more you learn. So, with that, coming to the conclusion of this module, we are at the end of the module molecular visualization. We have learned the journey of molecular visualization through the study of history of molecular visualization. We have seen how molecular visualization starts with the development of real life models made of metals, metal wares, wood, glass and other sculpturing methods. You have probably remember a very significant story of Byron bends. If you do not remember, Byron bend is type of wares made through machines which could be a low thickness bended wares, and two structures, one of an antibody and another of a disulfide mutase. The model for both of them was created, the physical model was created by Byron bends. And when both of the structure was arrived in the meeting, we have seen, we have read the experience of Dr. Richardson, who worked on, and to his surprise he find them with identical fold, and this is the first time when people have seen two sequences with 9% identity provide identical fold. So, this sculpturing objects not only help people to see the models, but also help developing new science. Then we have changed from the journey of sculpturing to the computer related models. We have studied the databases like PDB, PDB-E, PDB-J, the NMR database, the electron microscope database, the nucleoside database, all are merged together to global PDB now, and what type of files they have, the PDB file, the MMSIFT file, the MTZ file, the XML file, the FASTA file, all type of files and their importance we have studied. And then we have studied different softwares. We majorly divide them into two parts, one visualization software with modification, one other one only visualization platform to make good quality figure and movies, and we have chosen each representative one and one from them. The modification software Qt is the representative, and from the visualization Pymol is the representative, and with that we have learned how to study those softwares, how to know them, how to use them, and we end up making an example of how to make a high quality figure, publication quality figure. I hope all these things will help my young generation structural biologists to be excited to learn more through visualization. As I always believe, seeing is believing. So, you will see more, I expect you will be more excited, and with this I would have make the new generation more excited to the 3D structure of biological macromolecules. Thank you very much, you guys are wonderful in listening. As I always say, keep listening and keep asking us question if you have doubts. Thank you very much.